afternoon. I'm Ellie Walmark, and welcome to the ICM Research in Focus podcast series. Today I have with me Dr. Christopher Jones and Dr. Ashley Nicholls to talk about the association between radioresistance and tumour evolution and their work on using more complex tumour models to understand how tumours change over space and time in response to radiotherapy. Chris is a clinical lecturer at the CRUK Cambridge Centre at the University of Cambridge and also co-director of CRUK RadNet Cambridge. Ash is a research associate with the CRUK Cambridge Institute and CRUK RadNet Cambridge. Good afternoon, gentlemen. I know both of you are part of RadNet, as we've said, and Chris, obviously you co-lead it. So just to set the scene, first of all, can you tell me a bit about what RadNet is, please?
Yeah, certainly. So thanks for having us on today. So RadNet is a Cancer Research UK funded network of 7 centres across the United Kingdom that are dedicated towards doing radiation research. And it was a network that was set up really in recognition of the need to increase the amount of radiation research that we're doing to try and improve outcomes in cancer, and to do that effectively by centralising expertise and bringing together a pool of scientists and clinicians with an interest in radiotherapy as a means to overcoming some of the things that we're going to talk about today, like radioresistance.
Great. So first of all, just to start us off, can you both briefly outline your current research focus so that we know what you're working on at the minute? Do you want to go first, Chris?
Yeah, of course. Thank you. So my research focuses on improving the radiation-related outcomes in esophageal cancers. So for both esophageal adenocarcinoma, the glandular form of esophageal cancer, and esophageal squamous cell carcinoma, which is the more common form worldwide. And what I'm focusing on in particular is 2 things. Firstly, trying to find out why it is that these tumours are resistant to radiation, and importantly, why some patients with these cancers are more resistant than others. And that's when we really start to think about things like plasticity and evolution that we're going to talk about today. The second thing that I'm interested in, and we've done some work on before, is trying to find new ways to identify recurrence in patients who have been treated with radiotherapy. So one of the big problems that we have, particularly for the glandular form of this cancer, esophageal adenocarcinoma, is that it is quite resistant to radiation. So unfortunately, that means that quite a significant proportion of patients who receive this treatment will go on to have recurrent disease. But we do still have options for treating that recurrent disease if we identify it earlier. So what we're trying to do is to find new ways, whether through blood-based approaches, such as using ctDNA, circulating tumor DNA, or whether it's through using technology developed here in Cambridge by Professor Fitzgerald, including capsule sponge technology. That's something that you can swallow down and a mesh is pulled back and it collects cells through the gullet. And we're looking to see whether we can use those to identify recurrence earlier in those patients that have been treated with radiation overall, so that we can ensure that more patients are able to benefit from radiotherapy treatment.
Thanks. And Ash, tell us about your work and your research focus.
Okay, so hello, it's really nice to be here. So my background is primarily as a molecular biologist. So our lab mainly focuses on designing genetic tools, manipulating genes to investigate specific biological targets in cancer. So recently I started to work with the RadNet. Well, when I say relatively recently, within the last 3 or 4 years. So we've been applying some of these techniques that we have in the lab in the context of radiation to explore some of the resistant mechanisms which drive relapse and resistance in a subset of lung cancer patients. So one of the techniques that we use is something that we've termed wild-seq, which essentially allows you to track individual cells within a tumour as they're treated with radiation or any other drug, and it allows us to then look to see how patients respond or how tumours respond to the drug or the radiation. What we're really trying to pull apart here is which cells are resistant and which cells are actually sensitive to the treatment, and if we can try and target that resistant population.
Would you target them with another type of therapy or a different type of radiotherapy? How would you go about sort of working out how to treat them if they are resistant?
So that's the question, and that's, I think, something that we're trying to tease apart at the moment. We're really trying to understand why patients relapse, why patients are resistant to specific treatments, and if we can then try and work out why these cells are resistant, we can then design either whilst they're having treatment or by the end of their treatment or when they relapse, what this resistant mechanism is, and then try and target that with another drug. So that's really the aim of our work.
I think that last bit, Ash, is key, actually, because, of course, at the moment, we know that although radiotherapy is a treatment that we use in a significant proportion of patients with cancer and that contributes to a significant proportion of cures, we do still have high levels of resistance. And we've known for many years that we can overcome that radiation resistance by using chemotherapy. But of course, chemotherapy, whilst it is a good radiosensitizer, has lots of effects on the other cells in the body and leads to quite a lot of added toxicity. And it acts on the cells all of the way through patients' radiotherapy treatment, which is usually over multiple days of multiple weeks. What Ash and I are focused on is trying to find more targeted radiosensitizers, more targeted drugs that can act just on specific parts of cell pathways to ensure that we are focusing on those bits that are directly making cells resistant to radiation, but also importantly working out when is best to give those. Because it may be that actually, rather than giving them all of the way through treatment like we do at the moment, if we better understand how the cancers are evolving and changing in response to the multiple weeks of radiotherapy treatment, we might then be able to pick out an optimal time to give those more targeted drugs, or even the less targeted ones like chemotherapy. But it might mean that we're not needing to use them for so long.
Yeah, that makes sense. And so you're sort of reducing toxicity for the patient in other ways by specifically using them at the right time rather than just using them as a blanket cure or a blanket target. Yeah. So can we just go back to basics a little bit? So tell me, first of all, how important is radiotherapy to cancer care, do you think?
So I would say very important, but not just because I'm biased. So the evidence suggests that as many as 60%, 6 in 10, of patients who have a cancer diagnosis benefit from or should receive radiotherapy. And we know that radiotherapy achieves around or contributes to around 40%, 4 in 10, of all cancer cures. And to put that into context, we think that surgery probably contributes to around 5 in 10 cures. And by contrast, we know that chemotherapy contributes to around 1 in 10 cancer-related cures. And so we can see and we know that radiotherapy is a key treatment modality. So it's used in lots of patients, contributes to the cure of a high proportion of patients, but also is important in other settings. So we use radiotherapy commonly as a treatment to try and control cancers and to reduce symptoms from it. And it's useful from everything from improving a patient's swallow in a patient who has a diagnosis of a gullet of an oesophageal cancer to improving the pain related to spread of cancer to bone, for example.
And can it be used on all different types of cancer, or is it more effective on some? How do you use it?
Yeah, it's certainly more effective on some than others. So we wouldn't use it in the main for your hematological, for your blood cancers, although radiotherapy does have a role in lymphoma, and we tend to find that lymphomas are actually exquisitely sensitive to radiotherapy. But by and large, we'd use it in a majority of solid tissue cancers, but the extent to which we use it differs by cancer because they all have their own level of sensitivity and resistance to radiation. And of course, with the way that we give radiation at the moment, which is a beam from outside of the body, when we're thinking about the tumours that we can use it in, we have to think about the structures in the body that are around where that cancer is that we may damage with the radiation beam going through. And so that dictates to a certain extent which cancers we're more likely to use the treatment for. It's also worth saying that in the main, we would use radiotherapy in patients who have a localized cancer. So we tend to use it in patients for whom the cancer is not yet spread. although we increasingly do use radiotherapy as a treatment for what we call oligometastatic disease. So use it as a treatment in patients who have one or two sites where the cancer is spread to, where we can use the radiotherapy to ablate to get rid of the cancer at those sites.
Yeah, sure. And we've touched on it a bit already, but is it given before or after or in combination with other treatments?
Probably a bit of all three. So if I take my tumour sites, the one that I focus on, esophageal cancer as an example, depending on the particular type of esophageal cancer, we can use the radiotherapy ahead of surgery as a treatment to try and downstage the cancer and make it easier to cut out. Or in some cases, and particularly when patients are, for whatever reason, not able to have an operation, we would instead often consider using the radiation as the sole treatment, as the definitive treatment itself. But as I outlined before, what we know is that with esophageal cancers, as with many other cancers, we tend to find there are relatively high rates of resistance to radiation. And so we'll overcome those by combining the radiation with chemotherapy. And of course, radiation is a localized treatment. And so there's often also a need to use chemotherapy to try and mop up any cancer cells that might be elsewhere in the body.
Right, that makes sense. How do you personalise a patient's radiotherapy plan? Is that something that you do? Obviously, I'm from the Integrated Cancer Medicine Programme, so I'm all about personalising treatment. Tell me a bit about that.
Yeah, so I'd argue that radiotherapy is really the ultimate in personalised therapy. We can't give it unless we do personalise it. So what we would tend to do as your typical pathway for treating a patient who has cancer who's to receive radiotherapy is we would ask them to first come in for something that we call a mapping or a simulation CT scan. That's a scan that's performed in exactly the same position in which the patient will be treated with radiotherapy, and we line them up using small tattoos on the skin and lasers. And what we then do is we get that CT scan and we go through each slice of that CT scan and we work out where we want the radiotherapy beam to be directed and what we want it to avoid. And of course, that will be completely individual for each patient. And we'll then work with a broad team. One of the nice things about working in radiation oncology is that you work with a big team, and we'll work with physicists and dosimetrists and complex computer algorithms to work out how we can direct the dose of radiation that we want to hit that person's individual tumour, whilst avoiding the different parts of the body around it that we're less keen on hitting because it would lead to toxicities and longer term side effects. And then what we do to further personalise that treatment is we pick the right dose for that patient and we work out how we're going to spread that out over a number of weeks. And we also work out which particular, often chemotherapy, we want to give with the radiotherapy to best radiosensitise that patient's tumour and make sure their treatment works.
So it really is a case by case judgment, isn't it? You just deal with each particular case and anatomy, all of the things about a person to work out what's best for them and where to best target the treatment.
It's absolutely that. And I think what we have managed to make huge strides in doing over the last couple of decades is improving the technology that we use to deliver the radiation so that we can give increasingly accurate doses of radiation to particular parts of the body. The next step, I think, in terms of that battle and that quest to personalise the treatment is to work out how we can biologically optimise the treatment for each patient. And that's really where the research that Ash and I are doing comes in.
Great. So if we can bring Ash into the conversation, can you tell us, Ash, what does radioresistance mean? We'll start with the basics and then we'll get more complicated.
Sure. So I guess you could think of it in a kind of Darwinian selection kind of way, in terms of not all cells are equal. Not all cells within a tumor might actually be able to resist some specific type of treatment. So radiotherapy might kill most of the cells in the tumor, but some of them are going to survive, and that could be due to pre-existing or acquired genetic or epigenetic traits. And so this is genetic mutations, or it might be that something that is not genetic but associated with genes in terms of how they're regulated. So There's lots of different resistant mechanisms that could be in one type of cell inside a tumor and not in another. So really trying to unpick what these cells that can survive the radiation, the radio-resistant cells, how they're doing that. Maybe it's enhanced DNA repair, maybe it's some kind of metabolic rewiring, maybe they divide slightly slower so they're not affected by the radiation treatment, which relies on accumulation of DNA damage. All cells in a tumor are not created equal. Let's say it that way. So some will be resistant and some will be sensitive.
That's a really good way of putting it. I like that. And so why is it important then to study evolution? So you've touched on that a little bit, I guess, when trying to understand radio resistance. Is it to do with how the cells change over time, and that obviously has an effect on whether or not they're going to be sensitive? Is that what you mean by evolution?
Exactly. So obviously throughout a patient's journey through their treatment, this isn't a short period of time. That over the time of their treatment, we're talking about multiple months. And in that time, the dynamics, the microenvironment, the number of different types of cells within that tumor are going to change. And that might be from things that we try to intervene with, like drugs or radiotherapy, or it might just be the natural evolution of that tumor as well. So there's different processes that make the way that the tumor actually looks and the way that it behaves very different from the initial stages of tumor formation to the advanced stages and through therapy as well.
Yeah, so there are lots of different factors at play, aren't there? How are you studying radio resistance? Because this sounds like it's something that's very detailed and on a very small level. Am I right? And so tell me what the challenges are there.
So I think some of the developments that different research groups have produced over the past few years have really helped to zoom in and look at what kind of changes are going on in an individual level. So we've really been able to focus on what the interactions between individual cells within a tumor can actually affect the patient outcome. So being able to track cells in terms of putting in specific tags into different cells, which we have genetic barcodes, so we can integrate genetic, like a small tag into the genome of individual cells, and each cell will have a different tag. And so that then we can track through time how each one of those cells, because we know that our cells divide over time, so there'll be several cells that all have the same barcode within them, how each population of individual cells responds to a specific treatment. And then we can really hone in and try and look at what those specific pool of cells are actually doing within their genome and transcriptome to be able to understand what makes them different from the cells that couldn't survive the treatment. And they're the important ones because they're the ones that are going to create the problem for the patient in the future.
I mean, that is amazing. So I am right, it is at a tiny, tiny level, right? That's just amazing that you're studying single cells and tracking them. That's, yeah, mind-blowing, really.
But I mean, this is a field that has really exploded in the last kind of 5 or 6 years, where we've really been able to look at individual cells and whole genomes of individual cells, where we can really look to see what the differences are, not just between different tumors between patients, but also what we would call intratumoral heterogeneity within a patient. So and this will really help us to pull apart why one patient might respond to a specific treatment and another patient might not necessarily respond in the same way. And I think that will really help outcomes in the future by really honing in and characterizing what individual patients have within their tumor and how we would predict that they would respond. And I think to dovetail on that is the fact that we have a lot of integration of AI, and we have a lot of new tools where we can actually use all this, which we do produce a lot of data, but we can actually have a platform where we can actually integrate all this information into that platform to try and give us some indication of how we could predict that these patients would respond.
Amazing, amazing. And then, so how will the broader adoption of these systems bridge the gap between lab research and using radiotherapy in the clinic? Chris, do you want to take that one?
Yeah, I can do. So I think to take things back a step, I guess some of the challenges that we have in terms of modeling radiation responses are that we would generally give radiation or radiotherapy in the clinic over multiple weeks. So we would expect to give a patient who we were giving potentially curative treatment to anywhere between 5 to 7 weeks of treatment, and they would tend to have treatment on each weekday. So you're looking at anywhere between 25 to 35 consecutive days of radiotherapy treatment. And we treat patients to a high dose of radiation. And the problem with that is that it has conventionally been very difficult to model that. And so if you take the 2D cells that are stuck on plastic that we've used for decades in the lab, and you try to give them a high dose of radiation, and you try to do that in the short time period, the 5 to 7 weeks that we give when we treat patients in the clinic, it is almost impossible. And actually, most of the historical papers that have looked at trying to model and generate resistance in these cell lines have tended to give a certain dose, a small dose over a few days, and then let the cells recover. And then they've gone again and delivered treatment. And what Ash is talking about and outlining for us here is that we're trying to understand how tumours that are heterogeneous, that have lots of different cells within them, will change not just over space, but over time in response to those multiple treatments. And of course, when we give the treatment in the clinic, what we're doing is we're really stressing those cells out over that defined point of that time period of treatment. Whereas what we're doing with a stuck on plastic 2D cell line, if we're stopping the treatment and restarting it, is we're giving it a chance to rest, and we're not quite modeling what we're doing. And of course, the other challenge with those 2D cell lines is that they tend to just reflect one cell type or one particular cellular structure and background. And so what we're doing that we think is more adaptable to the clinic is trying to use more complex biomimetic models that firstly better recapitulate that heterogeneity, the diversity that we see across tumours, but that secondly also allow us to give much higher doses of radiation and to give those within a more clinically relevant time period. And that's what we've been doing in our lab. And so we've been using a panel of patient-derived organoids, which are complex multicellular 3D structures that we have shown can receive a much higher dose of radiation than the classical 2D cells. And what we think that allows us to do is to have a much more realistic overview of how a heterogeneous and diverse cancer is changing in response to that stress over a short period of time. And it allows us to then look at everything that Ash has described in terms of how the genome changes and how the transcriptome changes. And in particular, what we can do then is we can look to see whether the resistant tumour that we get at the end of a clinically relevant dose is formed because that tumour has selected particular cells that were always radiation resistant and have developed and grown in number as you've treated them, or whether we're actually, for one reason or another, creating new clones that are radiation resistant through delivering the radiotherapy, or whether it's a combination, or indeed whether you arrive at a point where you have pretty much the same clones that you had in your first tumour, but they've all developed epigenetic and different ways of responding to the radiation that makes them fitter and able to live through that radiation and form resistance. Then when we work out which one of those mechanisms or multiple of those mechanisms are contributing to the resistance, we can then identify the targets from them to make the cancers more sensitive.
That is a wonderful explanation. Thank you. So can you tell me a little bit about what are your key findings from the models that you've made? Where are you now in your thinking?
Just to follow on from what Chris was saying just a minute ago, we do find that depending on what the treatment is, because we don't just focus on radiation. We have some chemotherapy drugs as well, which we kind of use to select different resistant clones. And depending on the combinations of radiation and different drugs and how you give them, you can select out specific clones or different clones depending on how the treatment is given and in what order and what sequence it's given. So it's really interesting to kind of see the trajectory of different cells and resistant cells and how they look transcriptomically over time when you change the treatment. So I think we're kind of now starting to realize and unpick that it does matter in terms of what sequence or order you give a drug or you give a radiation treatment or what type of dose you give. And that will become a lot more important when we are trying to characterize and subcharacterize different patient groups to decide what kind of treatment to give them. So I think that's one of the biggest findings so far.
You've both used the word transcriptomics a couple of times, and I'm not sure that I understand fully what that means. Is anyone brave enough to give me a description of what that word actually means?
Yeah, sure. Yeah, sure. So I would say that transcriptomics is basically, although we always talk about genes, and these are pretty much stable things that exist within a cell, but the consequence of a gene is that it is converted into some kind of action within that cell. So it's going to have an outcome. So the gene will have an outcome. A transcript, when we talk about transcriptomes, we're talking about the intermediate between the gene and the outcome. So this is where the gene is basically being put into action, whether that's being requested to do more work or less work within the cell in the context of all the genes that you have within a cell. So when we say something is transcriptionally overexpressed, we mean that our cell is calling that gene to do more work. So if we have a gene that is very good at repairing DNA damage, and it is very good at asking the cell to recall that gene to do more work, as opposed to another cell which isn't good at doing that, then we would expect that cell to be resistant.
That makes sense. Chris, did you have anything you would add to that, or are we going to leave that one with Ash?
Cheers.
No, I agree with Ash. I guess one of the key things that Ash and I are trying to explore with this is whether over those multiple days and the multiple weeks of radiation, whether you are selecting for cells that are sat there and they've got a fixed change in their gene. So it might be a mutation in a particular gene that makes that gene more active and will always be more active. And if that active gene is then conferring on that cell resistance, it might well be that cells with that fixed active gene become more prevalent as the radiation goes on. The other way of thinking about it, though, is that, and particularly in cancers such as esophageal adenocarcinoma, which I act on, there's a very low rate of changes in genes that are fixed. And so you have quite a diverse pool of cells, but they don't have fixed changes in their genes. And so we wondered what would happen as we deliver radiation. And what we see, and this is where the transcriptomics comes in, is that we see that they adapt. And so they get changes in response to the radiation. And we think those changes are epigenetic. So fueled by the environment, by getting the radiation and switching on certain genes. And the cells are en masse managing to adapt, and they're changing which particular genes are active and producing the proteins that act as part of the cellular machinery for the cells and tell them what to do. And I think that's one of the things that we're most interested in with this, is that interplay between what we would describe as evolution of particular cells with particular fixed changes in their genes versus adaptation and what we call plasticity, as the cells sort of adapt to their environment and become fitter as a result. And certainly what we found predominantly through our research so far is more of the latter. We find that cells are plastic, they're managing to adapt to the radiation in ways that essentially make them en masse more resistant.
Right. That is amazing, isn't it? So, I mean, our bodies are amazing at healing ourselves, and they're also amazing at taking something like a cancer and making it grow quickly. So it's kind of just the way the body works. I get it. I think I get it now. Thank you.
I think so, and it also, when you think about it, it makes sense because our bodies have had to evolve to be resistant to radiation to a sense. There's that radiation in the atmosphere all the time. It's just in this particular context, we don't want that mechanism to be active.
Yeah, makes sense. Yeah. So, is your research being used in a clinical setting yet? Tell me where we are with that. Who wants to take that one? Chris, do you want to take that one?
Yeah, can do. So no, we're not in the clinical setting yet, but we are using tissues and samples from patients, which we're immensely grateful for and which really help us with our research. So we have within the Fitzgerald group in Cambridge a biobank of patient-derived organoids, and so those are those complex 3D models that are derived from tumour samples taken from patients with their consent, and we grow those in the lab, and they're a brilliant and very useful model for us to be able to study how tumours respond to radiation. And what we'd like to do is to be able to use some of the targets, potential targets for essentially sensitizing cancers to radiation that we've identified from our work. In time, we'd like to move those towards using them in the clinic, hopefully for patient benefit. What we're also doing alongside is we're not just using live cells that we're growing in, but we're using tissue samples from patients who've taken part in trials. So we're immensely fortunate to have funding from Cancer Research UK to use a cohort of samples from something called the Scope 2 trial, which is led from Cardiff University and Berlindra Cancer Centre with Professor Tom Crosby as the chief investigator. And this is a trial that's comparing standard and higher dose radiation in patients with esophageal adenocarcinoma and esophageal squamous cell carcinoma. And what we're doing is we're working with colleagues at the Institute of Cancer Research, University College London, the University of Birmingham, and Cardiff University to analyse tissue samples that were taken from some patients with their consent before they received radiotherapy and afterwards. And we're going to compare those two groups of patients, the patients who had the standard dose radiotherapy and the higher dose radiotherapy, to see whether the difference in the dose of radiation that was delivered in the same time period leads to a change in how those particular tumours evolved, both in terms of the cancer cells themselves, but also the stroma, the environment around the cancer cells that Ash outlined earlier.
Amazing. Ash, did you have anything you wanted to add in from a lung perspective?
Yeah, I mean, in terms of the research that we're doing in the lab at the moment, so as I said before, we are tracking how these tumours respond to different treatments, but the treatments that we're using and the different combinations that we're using are all clinically relevant doses, they're all clinically relevant regimes, and then they're being adjusted to look to see if we can optimize and make those treatments kind of more effective. So I think although we're not in the clinic with any of this at the moment, I think we're well positioned to be able to inform the clinicians on what might work best for specific cohorts of patients. And obviously we don't have the barrier of having to get FDA approval for new compounds or drugs so we're already looking at FDA approved drugs. So these are things that clinicians already use, and these are things that patients are already receiving. So it's more a case of it's more descriptive in terms of what these drugs are actually doing in the patients and how we can make that more effective.
Yeah, I see that. So what impact do you think or hope that your research will have on patient treatment outcomes in the future? Ash, do you want to take that one first?
So in terms of how much impact, I think you can already see that there's impact in personalised approaches, especially in breast cancer over the past 20 years. And I mean, they really are leading the way in determining what kind of treatment you should have for the genetic subtype of cancer you have. So there's a strong foundational understanding of the disease for breast cancer. And obviously there's a lot of money and work and time gone into that. And I think as we progress with other types of cancer and we get more data and we have wider patient populations in terms of a bit more diversity in ethnicity and just in terms of variation in patient sampling and better patient storage, and because obviously we're taking samples, we've got historical samples from patients, but we've got a lot of techniques now that require different storage methods for those biopsies. So I think in the next 5 or 10 years, we'll have better sample quality where we'll be able to do more techniques that will give us a lot more information on specific diseases.
It's almost like you have to catch up. The technology moves at such a rate that you have to catch up with the sampling, don't you, as it goes? And it's so hard to predict what might happen or might be required in the future. So you're always trying to play catch up, I guess.
And this is something that's super important, especially in radiation. There's a very limited number of normal samples. So when I say normal samples, I mean patient tissue that is not cancerous, because that is super important for determining how we try and spare normal tissue tox, how we kind of decide what the difference is between something that is responding and isn't responding in a cancer specific way. So as we are now learning that it's super important to be able to capture a patient's normal tissue as well as the tumor tissue as well, which wasn't really the main focus maybe in the last 40 years of cancer research.
Chris, do you want to tell me about your impact on treatment outcomes, what you hope will happen?
So I think this field as a whole has a huge amount to contribute to improving outcomes in radiotherapy. So to be pessimistic for a minute, radiotherapy is far behind where systemic therapy is in terms of being able to biologically personalise our treatment. So despite very strong efforts from the community over the past couple of decades, we've really struggled to identify the particular molecular changes in cells that would either allow us to predict who is more or less likely to respond to radiation, so good biomarkers, and also, as we've discussed, identify those changes that are contributing to resistance so that we can find kinder and more effective drugs to be able to personalise and radiosensitize the patient's treatment. Where I think we can go by looking at the evolution, plasticity angle and the work that Ash and I described is that we, hopefully by understanding how cancers change in response to radiotherapy, will be in a much better position to find the particular cellular changes, the molecules that tell us those who are most likely to respond. So we do find those biomarkers. And equally, I think we'll hopefully be in a position where we can finally begin to identify some sensitive and effective targeted radiosensitizers. And to put that into more context, it's now been a couple of decades really since the last FDA approval of a targeted radiosensitizer to be used in any tumor site managed with radiotherapy. And if you compare that with the multiple approvals there'll be each year for targeted drugs otherwise outside of the radiotherapy setting, that really shows how behind we've got. But I think a lot of that is actually based on models that in the past were, because of poor technology, and inadequate. Whereas I think we're now going to be in a position where we can use more complex biomimetic models, such as the organoids we've described and the epithelioids we have in Cambridge, led by David Fernandez Antron. But in addition to that, the more complex ways of analysing them. And I think that will leave us in a much better position to be able to identify those biomarkers and hopefully new targets for drug sensitizers.
Amazing. So my last question really is, where do you two see yourself in the next 5 to 10 years? Where do you see your research taking you? I'm hoping you'll still be continuing, because it sounds just completely amazing what you're doing now. So yeah, where are you going to be in the next 5 to 10 years? Who wants to go first? Chris, tell us your thoughts and then we'll give the last word to Ash.
Yeah, thank you. So we're going to carry on, I hope, trying to find out what the causes are and how cancers evolve under the pressure of radiation. And we're going to carry on doing that in the context of esophageal cancer. And I hope that what we'll do over the next 5 or 10 years is find some good targets for drugs. We already think we have some, and then begin to roll those through to being used in patients. What we're also keen to do is to better understand how different forms of radiation can affect how tumours change over time. So one of the things that we haven't discussed today, but that we're getting very interested in, is the difference between the way that we've conventionally delivered radiotherapy, which is in those multiple daily treatments of high dose rate external beam radiotherapy, how that might differ to the way that we are likely to be moving to deliver at least some of our anti-cancer radiation, which is in the form of targeted radionuclide therapy, which is essentially radioactive drugs, so radiation we can inject into the bloodstream and deliver to tumours. And that's a very different type of radiation. It's a lower dose rate and it's a bit more variable across a tumour. And we're very interested in how that would impact on how a tumour evolves and develops plasticity in response to radiation, and how that would differ from the conventional high dose radiation. And alongside, what we're hoping to do is that bit I outlined at the beginning, which is not just to make radiation therapy more effective, but also be able to use it in more patients as a kinder treatment for patients by beginning to use the technologies that we have here in Cambridge, such as relating to circulating tumour DNA or the capsule sponge, to be able to better identify recurrence and identify recurrence earlier in patients who've received radiation, so that we can broaden the pool of patients who are having more effective treatment.
Thanks, Chris. And that all sounds amazing. And Ash, tell us where you want to be in the next 5 to 10 years.
I mean, I was really hoping that this research would lead to a specific type of rum in Barbados to be a radiation sensitizer, and maybe I could head out there to do some really in-depth research. But
I mean, that sounds great, no?
I mean, unfortunately, I can't swing that one at the moment. But so I'm going to put that 5 to 10 year plan on hold to open a rum shop in Barbados. But in the meantime, maybe in the next 5 years instead, there's really 3 kind of aspects that we'd like to develop and kind of build on that we have at the moment within the RadNet group. And so from my point of view, we'd obviously like to develop this lineage tracing kind of wild-seq platform where we can look at different clones and how they evolve and how they change and adapt over time. So what we would like to do, and like I said at the start of this podcast, we are developing different genetic tools to try and kind of answer questions in cancer research. So we're in the process of developing a new tool at the moment where we can actually select out those resistant clones within a tumor before they've had treatment. So we can identify which clones are going to be resistant already, like at the start, before we've even treated, take those out, and then be able to treat the tumor and see if we can actually give some kind of curative dose of radiation drug combination. So that's one of the aspirations in the short term. One of the other companies that we've been working with at the moment is a company called Okamura. So they're a small biotech startup in Paris. And what they actually do for breast cancer patients is they take a small biopsy, they create a organoid or 3D model of that specific tumor, and then they give a range of different standard drugs that most of these patients would get anyway, but in different combinations, and then look to see which organoid responds the best, and then they feed that back into the kind of clinical workflow to help the clinician decide which combination of drugs they should be on. Now, we'd like to implement this into a kind of radiation drug combination workflow. So that's something that we're trying to work up at the moment as well. And just finally, quite a lot of people have heard about CRISPR screening and CRISPR, and essentially this is knocking out genes or stopping the function of genes so that we can work out what their influence is on the response to a specific drug or treatment. And so although we can do this very successfully for the whole genome in 2D on plastic, and some people have done this in organoids, it's very difficult to be able to run a genome-wide CRISPR screen. So being able to screen all the genes within the genome in a live animal or obviously we wouldn't do this in patients, but within in vivo models. So we're now currently working on trying to be able to run in vivo CRISPR screens genome wide. So they're kind of next 5, 7, 10, 15-year plan.
Amazing. Thank you both so much for taking the time to talk to me this afternoon. It's been such an interesting conversation. I feel I've learned a lot. I hope my listeners have too. I really appreciate your time. Thank you, gentlemen.
Great to
Thanks
be
for
here.
having us.
Thank you. Thank you.